Welcome to A-Level Biology, Transporting Animals. This is the MCQ Physics and Maths Tutor Pack. It's a walkthrough. See the link in the description for access to the questions and mark scheme. This is really for OCR students, but it's also relevant to AQA students. So which of the options A to D best describes the chloride shift? These are all multiple choice. What I'm going to do here is draw what's happening roughly in the chloride shift. We've got carbon dioxide joining with water to form carbonic acid. This then breaks down into hydrogen carbonate ions, HCO3 minus, and hydrogen ions, H plus. We then get chloride ions shifting in to balance things up, and this is known as the chloride shift. So for A, hydrogen carbonate ions and chloride ions both move into the red blood cell. Well, that's patently untrue here. One's moving in, one's moving out. What's actually happening is chloride ions are moving in and hydrogen carbonate are moving out. So B is the correct answer. C and D are also incorrect. Carbonic anhydrase doesn't join with chloride ions to produce carbonic acid. That was carbon dioxide and water. Okay, let's have a little look here. Question 2. The ECG traces below show 4 abnormal heartbeats recorded for 6 seconds. Which of the traces shows atrial fibrillation? Well, we can see B's ectopic, highlighted in blue here with the missed heartbeat. We can see that C looks fairly normal, but probably a little bit slow. So that's bradycardia. I've only written Brady because I'm lazy. And then the bottom one's fibrillation, where it's kind of all over the place. So that seems like it could be a possible answer. But we're looking at atrial fibrillation. See what I've circled there? That's the P wave. Okay, those are the P waves. Let's zoom in a little bit closer to show why A is the correct answer. So if you look here, we should have PQRST, but what do you notice about the P wave? We've got a double wave. It should be just one wave. This means we have fibrillation of the atria. Okay, so they're getting extra contractions and all out of time. So we've got fibrillation of the atria. There we go. That one was a little tougher. Okay, question 3. The sounds of the heartbeat due to the heart valves closing can be described as lub-dub. Lub sounds occur at the beginning of the ventricular systole. Which of the A to D describes what happens when the lub sound is heard? So we've got 4 options here. Correct answer is it's the AV valves, and essentially when they close, the blood splashes back against the atrioventricular valves, both the bicuspid and tricuspid. So sound of the AV valves closing, or really blood splashing back against them. So your answer to this one is D. Of course, the dub is the sound of the semilunar valves closing and the blood flowing from the aorta and pulmonary artery falling back against them. Let's move on to question 4. Which of the options A to D on the diagram below shows the time at which the SAN sounds out a wave to initiate a heartbeat? Okay, well, let's look a little bit into this. The sinoatrial node sends out a depolarizing wave or wave of depolarization, an impulse across the atria, causing the atria to contract. So I'm looking for somewhere where atricular or atrial pressure is greater than ventricular pressure. So you can see here at B, atrial pressure, which is shown by the large dashed line, is bigger than the small dashed line, which is the ventricular pressure. And that suggests to me that the atrium are contracting, the volume's getting smaller, and we have contraction of the atria at B. Okay, let's look at question 5. So, which of the options A to D would result in the formation of tissue fluid? We have A, hydrostatic pressure is less than oncotic. Hydrostatic pressure equals oncotic. Oncotic pressure is below hydrostatic pressure. Oncotic pressure is greater than hydrostatic. Well, the correct answer here is C, and I'll explain why. So, at the arterial end of the capillary, the hydrostatic pressure is larger than the oncotic pressure. So the sort of pressure created by osmosis into the capillary is less than the hydrostatic pressure pushing out. And this means we get a formation of small things leaving the capillary, forming the tissue fluid, things like water, glucose, and other small molecules. So C is our answer. The formation of the fluid at the A arterial end is when the hydrostatic pressure is greater than the oncotic pressure. Let's move on to question 6. Blood vessels are adaptive for their function. Which of the following statements are true? The walls of the arteries near the heart contain a lot of elastic fiber so they can stretch and recoil to maintain blood pressure. Well, we know that's true. You need elastic in the aorta because the pressure is so high. The walls of the venules contain little muscle. That's true. Veins or venules contain hardly any muscle. The walls of the arteries contain a lot of muscle to contract and generate pressure in the blood. No, it's the actual left ventricle that contracts and generates the pressure in the blood, not the muscles within the artery that contribute to pressure generation. So the answer is B, 1 and 2 only. Hope you got that right. Make sure you have a go at the pack, or you pause the video and have a go at these before I run through the answers. Which of the statements A to D correctly explains why tissue fluid forms in plasma membranes? Well, we just did this. The hydrostatic pressure must be greater than the oncotic pressure. So H must be bigger than the osmotic pressure, which is created by the pressure of proteins in the capillary, which we call oncotic pressure. Okay, 8. Let's get into this one. The hormone ecdysone is synthesized in the prothoracic glands found in the upper thorax of some of the invertebrates and is released into the hemolymph. It is then transported near the cell surface body and causes the loss of the exoskeleton. So this is kind of how an insect sheds its exoskeleton. Which of the following statements explains this? Well, it's synthesized by special neurosecretory cells. Well, we have no evidence for that. However, it must be soluble because it tells us that it's transported um, in the hemolymph. So it's soluble, and that means it's probably a polar molecule. So 2 is correct. Also, it's complementary to receptors on the body cells. Well, we're also told that it acts near the cell surface, so it probably is complementary to body cells. So we want the answer that is 2 or 3 only, and that is C. There we go. Okay, question 10. Let's go. Which of the statements A to D explains why diastole follows systole in the mammalian heart? Well, is it because the cardiac muscle is myogenic? That means it self generates its own impulse. No, that's not the reason. The reason why distole follows systole is because we need a period of relaxation after contraction. It takes time for the muscle to repolarize and it can't contract again until that has been completed. The D, SAN, receives impulses from the AVN. That's the other way around, so that one's wrong. Right, 10. The diagram below shows the internal view of a heart with the atria removed, so we can just see the valves. Where does oxygenated blood push through these valves when it enters a ventricle? Well, oxygenated blood returns to the heart through the pulmonary vein from the lungs, and it pushes through the bicuspid valve, which must be represented by C, as there's 2 parts, into the ventricle. Whereas B is the tricuspid, that lies between the right atria and the right ventricle. And we're dealing with oxygenated blood here, so we're looking at left atria to left ventricle, and remember it's the bicuspid, that is the AV valves in between those 2. Super. Answer C. Remember to pause the video before attempting any of these questions. It's a good idea. So, 11. Myoglobin is a protein found in muscle. It has a very high affinity for oxygen at the most partial pressures of oxygen. The figure below shows the dissociation curves for an adult hemoglobin, fetal hemoglobin, and myoglobin. Which of which of the rows A to D shows the correct labels for the lines? Well, if we look at X, you can see has the highest saturation of oxygen, even down to low partial pressures. So that must be myoglobin. That has very high affinity for oxygen. Now, fetal hemoglobin must be Y. Why is that? Haha. Why? Because fetuses, feti, whatever the hell they're called, live in a low oxygen environment. Their hemoglobin has to have a high affinity for oxygen. They're not very active, so they don't need to unload the oxygen readily, as they're not like doing ultra respiration and pumping up their muscles. But they do need to hold on to oxygen because inside the womb is a low oxygen environment. So they have a higher affinity for oxygen than your adult hemoglobin, which must be Z. So we are looking at the answer A. X is myoglobin, Y is fetal hemoglobin, and Z is adult hemoglobin. You'll find things like lugworms also, and creatures that live in low oxygen environments. They have a sigmoid curve pushed to the left of adult hemoglobin, or human hemoglobin. Let's move on to question 12. The table below shows the different percentages of 3 components of blood vessels. We've got elastin, smooth muscle, collagen. Which of the rows A to D shows the proportions of the components of the aorta? That's a huge artery. It needs loads of elastic for recoil. So you're looking at C or D really, because it needs tons of elastic to stop it snapping. And it needs collagen for repair and strength. and it doesn't need as much smooth muscle quite as 45%. Right, 13. The trace below is an electrocardiogram, ECG, of an abnormal heart activity. What is the name for this? We've seen this one on some of the earlier ones. You can see here that this is going to be an ectopic heartbeat because we've got some heartbeats missed. If it was bad boy bradycardia, it'd be a lot slower than this. But you can see here where I've circled in red, we've got some parts of the heartbeat that are missing. And it's not tachycardia, it'd be a lot faster. And fibrillation would be all over the place, no recognizable areas. Okay, let's look at 14. The graph below shows the oxygen dissociation curve for adult hemoglobin. So here's the sigmoid curve. We've got saturation of oxygen on the left and PO2, partial pressure of oxygen, at the bottom. What is the proportion of oxygen molecules released by hemoglobin between 40 and 20 milligrams? So you just draw a line up at 40, zang a line across to your oxygen sats. That looks about 78, 77. I'm just doing this roughly to get the right answer because it's multiple choice. So I don't need it exact. I just need closest to. And then if I zing up from 20 milligrams of mercury or partial pressure, I zing across and I'm going to get like, what is that, 24? I don't know. 22. Yeah, 22. So 77 to 22. So the difference between them is 55. We know that the starting saturation was 77. So I can do the difference over the original, and that will give me the proportion that has been unloaded. So what is the proportion of 55 out of 77? It's roughly 0.7. So it looks like my answer is going to be B. I know that wasn't exact, but I'd need to zoom in on that graph to get the little squares perfect for that. And that's close enough for me to confirm the answer is B. Awesome. Let's move on.
Keep moving on, keep moving on, keep moving on, keep moving on.
15. Carbon dioxide released during respiration can affect the percentage of oxygen saturation of hemoglobin. The tertiary structure of hemoglobin is affected when CO2 reacts with water to form carbonic acid. The reaction releases hydrogen ions. Which of the statements explains the shape change? The release of hydrogen ions causes the pH to rise. Hydrogen ions lower pH because they're more acid. You know that from GCSE chemistry. That makes A and B wrong. The release of hydrogen ions causes the pH to fall. Now that is correct, which increases hemoglobin's affinity for oxygen or decreases its affinity for oxygen. Well, the answer is D, and the reason is because during respiration, you want a low affinity for oxygen. You want your hemoglobin to release oxygen so your muscles can use it for respiration. The last thing we want is our hemoglobin to be hanging on tightly to the oxygen and have a high affinity and not release it or unload or dissociate the oxygen into the working muscles. So during respiration, the change in the hemoglobin makes the hemoglobin have a lower affinity for oxygen, releasing it more readily for respiration. Right, 16. The aquatic crustacean Daphnia magna has a heart that pumps blood, a blood-like liquid called hemolymph, around the body cavity. You'll find a lot of insects have that. Which of the statements A to D describes the circulatory system of Daphnia magna? Well, it's single. They don't have lungs, so it doesn't need to go back to the heart, get re-energized, blah, blah, blah. It's not a double circulatory system like our heart. Is it closed or open? Well, closed means that the blood is held within veins and arteries. Do insects have veins and arteries? No, they don't. They don't have capillaries, veins, or arteries. So the Daphnia has a single and open system. The answer is B. Let's move on to 17. A student studied the structure of a blood vessel and found an outer layer of collagen fibers followed by a thick middle layer of smooth muscle and elastic tissue. Sound familiar? And an innermost layer of endothelial cells. Well, when you draw it out and sketch it out as it's said there, you know what it is. It must be an artery, okay? It's got a small lumen, really thick walls made of elastic and muscle. So the answer is A, artery. Okay, let's look at the next question. 18. In the graph below, the top electrocardiogram ECG trace shows the normal heart activity and the ECG trace below shows an abnormal one. What is the heart condition represented by this ECG wave? Well, we know the one at the bottom must be fibrillation when it's absolutely all over the place. Tachycardia means faster, brady slower, and ectopic is when you miss part of the wave. 18. Which of the options A to D 19, is the correct statement about tissue fluid? Tissue fluid carries carbon dioxide to muscles. Nope. Oncotic pressure in the capillary causes tissue fluid to formation from the plasma. Hydrostatic pressure in the capillary causes tissue fluid formation from the plasma. And yeah, it's C. Purely because, remember, at the arterial end, the hydrostatic pressure is greater than the oncotic pressure. So small molecules like water, glucose, ions are forced out of the capillary, forming tissue fluid. 20. Which of the following statements correctly describes the mechanism behind the water movement between plasma and tissue fluid in the venial end of the capillary? Right, this is the different end of the capillary. So at the end of the capillary bed, we have the venial end. And at that end, the hydrostatic pressure has fallen. That is pushing out from inside the capillary to out. So at the arterial end, it's big here where I'm drawing it now. But at the venial end, the blue end on my diagram, the hydrostatic pressure has fallen. It's fallen so much that water moves back into the capillary by osmosis. And so the oncotic pressure is greater than the hydrostatic pressure, so water moves into the capillary. The answer is D. Let's look at 21. When you listen to a human heartbeat through a stethoscope, you can hear a stage lub-dub sound. Which of the following causes the lub? We already covered this one. The lub is the sound of the AV valves closing, the atrioventricular valves. 22. The following events occur when carbon dioxide enters an erythrocyte, a red blood cell, in a capillary. So we need to get this in the right order. I would say just draw this out each time. Carbon dioxide and water combine to form carbonic acid. They then break down to form hydrogen carbonate ions and hydrogen ions. The hydrogen carbonate ions shift out of the red blood cell and chloride ions shift in. So we want to put this in the correct order. And our correct order, CO2 reacts with water to form carbonic acid. So 3 is first. Carbonic acid dissociates would be next. Hydrogen carbonate ions diffuse out. And then chloride ions diffuse in. So we're looking at B for this one. B is the correct answer. Hopefully you got that right. I think there's one more question to go, or is that it? Okay, final question, 23. Dissolved material gives rise to oncotic pressure, which is related to water potential. Which of the following shows the typical oncotic and hydrostatic pressure in the blood at the arterial and the venous end? Well, at the arterial end, hydrostatic is bigger than oncotic. So we're looking at, it has to be C or D. And then if we look at the venous end, we can see that the hydrostatic pressure must be lower than the oncotic. So again, is C or D. The reason C can't be right is because they're showing the oncotic pressure changing from 20 to minus 20. The oncotic pressure doesn't change. It's the hydrostatic pressure that changes. Why does oncotic not change? Because oncotic is created by the proteins, and the number of proteins in the capillary stay the same because they don't move out. So the oncotic pressure stays the same. It's the loss of fluid at the arterial end which reduces the hydrostatic pressure by the time it reaches the venial end. All right, let's go. Hit the light.
If you push the button,
Like me.
Would you like it?
You're still here? It's over.